My name is Kerry Hair, and I am a core research technologist within the Penn State University's Genomics core facility. I'm supported by the Huck Institutes of the Life Sciences, which is an institute, not a college, inside the university. And I am part of one of 11 core facilities that the university supports. I am in next generation sequencing, largely on Illumina platforms, and I specifically specialize in RNA and single cell sequencing. The things that we struggle with is overamplification. Overamplification is mostly an issue for our Amplicon PCR sequencing customers. We have a large microbiome group here at the university. They're called One Health Microbiome, excuse me. And some of the leading international experts in microbiomes work here and work with them. The main way that researchers have evaluated communities in microbiomes is through Amplicon PCR in next generation sequencing. Again, that's a ribosomal RNA amplification. There's a ton of overlap in sequence, so really low diversity samples and potentially really small populations of samples per species. And so overamplification really hides the diversity of species that they can see. So we've been concerned about overamplification in our Amplicon PCR work. And so when we learned about the iconPCR, we were very excited about using it to really target the optimum cycle number for each of our Amplicon customers individually. We started with the second part of the experiment first, which is the procedure that we use is a two-step. The customer uses degenerate primers on their target samples first. They do some currently unknown number of cycles, and then they pass it off to us, and we amplify with the Illumina adapters and indexes for sequencing. So we're currently targeting a minimum cycle amplification on that second half. And we've shown really positive results. What we've liked so far is just the ability to not only minimize cycle number, but also actually target it project by project. That's been really key. And sample by sample. And you can see that in some of the work that we've done. There are samples that really are low abundance. So they take a lot more cycles than everything else. And that's been really, really exciting to see that clear observation that we're seeing those differences. The other thing that we're really excited about is cost reduction by incorporating the iconPCR. We can remove several of the QC steps that are pretty expensive. We can remove those and or minimize them compared to our original SOP. For example, we would run every sample on a CyberSafe gel, an e-gel, before trying to amplify it. And we would do that afterwards as well. So if you have a plate of 96 samples, frequently we'll have multiple plates of 96 samples. That's 1, 2, 5 e-gels that you're doing, and then you're doubling that. So let's say 10 e-gels. So that's a huge cost savings that we don't have to do. And then additionally, the bead normalization that we would do, we would typically do the normalization on a sequel prep plate. Those are very expensive. And so having the opportunity to do the normalization in real time saves that cost as well. We estimate that once we fully incorporate this procedure, we will have reduced our overall costs by about 50% for an Amplicon project. The cost of the library prep by about half. It also reduces labor time, and labor is actually the most expensive. So reduces hands-on time, which really reduces labor cost. The use of the iconPCR has really opened my eyes to the different ways that these reactions behave, and I would use the specific example of the indexes, and I have. We have worked together and observed lots of different index-specific challenges, and that is one thing that has carried forward, where we've been able to say, this index, when we use it in a reaction, a pooled reaction, doesn't always work as well as we expect it to. Certainly there's material there, certainly it's we're getting enough reads, but it's not working as well as the others. And that has been really eye-opening, kind of peering behind the curtain, I would say, giving kind of a glimpse at maybe not necessarily trusting the chemistry as implicitly as we had been. And that's been a really fascinating experience. I don't think that it will necessarily impact our sequencing long term, but I will say we've noticed that certain plates of indexes work more reliably than others, and that you can see that in real time when you pool a bunch of samples together. The samples that we know to be tagged with less perfect indexes won't give you quite the number of reads that you might expect, and so that's been interesting, and makes me feel better about my pool balance, right? Like, as we incorporate it into the SOP, it has made some really dramatic changes, and I would characterize those as financial changes. So the ability to eliminate some of those QC steps that are costly, the ability to go from, and then on the labor side, the ability to eliminate some of those QC steps, really reducing labor hours. My long-term hope is for the Amplicon work is, and this is a little bit off topic, but I wanted to make sure I shared this, is that we hope to really incorporate the icon into both parts of the experiment rather than just the second half. So one of the really interesting things that I foresee is being able to use those to generate primers against our customer samples, and minimize those cycles, as well as the second step with the Illumina adapters and primers. I think that's really going to shift the microbiome paradigm, like, fundamentally. And I'm really excited about that, because I suspect that we are missing things. And it'll be interesting to see how the research community takes that. We get into, I think as scientists, we get into ruts, and we, especially in academia, we've done it this way, so this has to be the way we do it. And the iconPCR creates these opportunities to really fundamentally rethink how we process these samples, and what our options are to see these microbiome communities in particular. What I see coming out of research laboratories can be, signals that are absolutely blown out of the water. Well, what are we missing in that first round as well? So I'm really hopeful that we can also kind of shift into again that degenerate primer amplification optimization, and then see where that takes us. I'm excited about what we'll find. This is a case where, like, the devil is in the details, and overcycling. Is it a huge problem? Maybe not, but scientific discoveries are frequently made at the margins, and if we're trying to get the best answer that we can, the most detailed answer that we can, we really need to think about those margins. And I think that we hide those details with overamplification. Are we still going to be able to build a transcriptome? Sure. Are we going to be able to sequence a newly discovered species? Absolutely. But it's the small details that really help us to understand how organisms function, and the ways that these, like, incredible biochemistries interact. It's in the really the fine tuning and the tiny differences that we can extract with our next generation sequencing. And so I feel that that's where the iconPCR shines, is because it has finally become easier to do that, easier to optimize those reactions and capture those details that might be missing in overamplified samples. So we use qPCR all the time to quantitate our libraries. And it wasn't until I appreciated that, I'm diluting those libraries by 10,000 to look at them on a dye-based qPCR. So it wasn't until I made that connection that I began to appreciate how little I needed to put into the icon reactions. And by that, I just mean, it was again, we're talking about a paradigm shift. Like, endpoint PCR's goal is to put as much in there to get a signal. So you're optimizing on the back end. And I had to kind of rewire my thought process to say, no, I need to optimize on the front end. I need to think about what is the maybe what's the minimum that I need to put in versus how much can I get out on the back end. And once I made that connection, things were a lot easier. Things fell into place.